So good night to everyone. Welcome to the SBF webinar. Today we have the pleasure to introduce Francisca Guzmán-Lastra from Universidad de Chile. She works at the Departamento de Física, Faculdade de Ciências, and she has made a doctorate in fluid dynamics and a postdoc in Düsseldorf in magnetic active particles. So, Francisca, you have some 50 minutes to tell us about your work.
Thank you, Leonardo. So, good evening, everyone there. So today I'm gonna tell you the story about the active carpets, and we will focus in active carpets in floating viscous films. This is the fourth work related with active carpets that I have been involved. We started with this new framework, let's say, to study biogenic transport induced by microswimmers in aquatic environments. We started this like 5 years ago, more or less, with Arnold Matthijssen in the University of Pennsylvania and Hartmut Leuben at Heinrich Heine University in Düsseldorf, Germany. And now to this active carpets research, I have the pleasure to have new colleagues involved in these studies. So I will present you a work related with the where the researcher Hugo Ulloa has been also involved, also my student Felipe Barros, and Professor Rodrigo Soto, and also the student Gabriela Aguayo. So let's start. So first, I will talk through all this presentation about microswimmers.
You should you should share your screen.
I think I was sharing my screen. Wait.
No, not yet.
Okay. Okay, here. Now you can see it?
Yes.
Okay.
That's
great.
fine.
Yeah, okay, let's go. Yes, so I will talk a lot about microswimmers. So for me, microswimmers are flagellated microorganisms that they are moving in a 2D surface, and they can be considered as dipole forces, like from the physical point of view. So they are dipole forces in the sense that they exert two forces into the fluid that we can separate in two different, let's say, regimes. So when the dipole forces are pointing in this direction and they are in the same magnitude but pointing in like outward the body of the microswimmer, we can consider that this microswimmer is a pusher-like microswimmer. And examples in nature of this kind of swimmers that exert this kind of flow fields, like in the blue arrows, are bacteria. So for us, when we consider a bacteria swimming in a fluid, for us as a physicist, is a pusher or a dipole force, okay? The flow field that you are seeing in blue arrows represent the far field approximation for a dipole force. So in the sense that if you want to measure anything with this consideration or this flow field, you must be far from the swimmer, okay? So if you want to measure something close to the microswimmer, then you have to add more multiples flows from the multiple expansion, okay? So you need, you have to consider another contributions in the flow field in order to solve something close to them. Okay, so if in another case, so if your microswimmer is now like a microalgae, such as the Chlamydomonas reinderdii, the dipole force is now pointing in this direction. So we have 2-point forces, which are in the same magnitude, but now they are pointing inside the body, okay, of the microswimmer. And then we have like a reversal of the flow field exerted in this case. So you can see that again, the blue arrows represent the flow field exerted by this microswimmer. And you can see that the flow fields are more like similar, but they are inverted. So in one case, the flow field is pushing flow from the head and the tail, and in the other case, it's exerting the flow from the sides. Okay? So for us, microswimmers are represented by a longitude, a length scale, which is in most of the cases the size of the microswimmer. So here I'm showing that microalgae in general is larger than bacteria, and they are also faster. So for us, the important physical parameters to describe these microswimmers are a length scale, also the velocity at which they are moving, that we consider it as a constant velocity. And you can see that also microalgae are faster than bacteria, and they are also larger, okay? And even though that they are like the microalgae is moving fast, we can consider them moving in a low Reynolds number regime. And in this sense, we can consider that the flow that they are exerting is in a very viscous limit. So there is no inertia and there's no time dependence in the flow field. So all the flow fields that these microswimmers are exerting into the flow are instantaneous, okay? Okay, so now let's continue thinking about these microswimmers. So now we know how we want to model them. We know what are the important parameters to describe them. So now we will think what happens when we put these microswimmers now moving close to boundaries, okay? So microswimmers, when they are swimming close to boundaries or interfaces, they are attracted to them. This attraction might be understood or can be understood from different point of views. So the first one is an hydrodynamic response, in the sense that when microswimmers are swimming close to a non-slip boundary or to a free surface, it has been observed that they perform this circle swimming close to the surface, and this circle swimming in turns generate long exploration times in the surface, and therefore an hydrodynamic attraction or a surface accumulation in the boundaries. So in this work of Lauga, they measure the density of microswimmers close to two non-slip boundaries. So one boundary was close to 0 and the other one was close to 100 microns. And you can see that the density of microswimmers is higher closer to these boundaries. Okay? So this is the first thing. So they exert long times exploring the surfaces or the interfaces, and they are also hydrodynamically attracted to them. The second contribution in the case of microalgae might be the phototaxis. So microalgae, when they are swimming close to free surfaces, they tend to accumulate closer to the free surface or where the light is higher, okay? So we can say, okay, they are also attracted to the free surfaces due to phototaxis in the case of microalgae. And there is another factor, which is the activity. So we know that microswimmers, when they move close to boundaries, or they are like inside a microdroplet, like here in this video, we can see that microswimmers tend to accumulate in the interface between these two fluids. Okay, and this accumulation can be also understood as a response or due to the activity of the microswimmers. So they get close to the interface, and since they are performing this run and tumble motion, it's harder for them to escape from the boundaries. So this is the simplest way to see that microswimmers are accumulating close to an interface, okay? So since they accumulate and they can cover large area, forming these floating biofilms, we are wondering how this accumulation of microswimmers can affect their environment. So in particular, we are interested in answer how the transport or the biogenic transport induced by these formations or this accumulation is changing the environment. So one question might be, is this fluctuations that they are exerting into the flow to the fluid is larger or smaller than Brownian transport? Or okay, so this is our motivation. So that is the case when we are swimming close to a surface, right? Or to a free surface or to an interface. So we also know that there are mechanical responses for microswimmers to accumulate close to fluid-fluid interfaces. Okay, so here, for example, we have different accumulation of microswimmers at fluid-fluid interfaces. And this accumulation might be due to straining or in situ growth or buoyancy, among others. So we can also consider, okay, so now we can be not super close to the free surface, but we can be now like a little deeper and close to a fluid-fluid interface. And we will see still this accumulation of microswimmers in these areas. Okay, so we also know that microswimmers are not alone in aquatic environments, and we also know that they need to get food from their environment. So we made this question of how, like, a large accumulation of microswimmers can attract or move or renewal nutrients close to them, like in the sense to increase their options to live or to reproduce there, okay? So what we did in this work here is to consider a model that for us it's called an active carpet, which is this black area here, which is full of microswimmers, okay? All the microswimmers are moving in 2D above this black square here. And what we did is we allow passive particles sedimenting. These particles were not Brownian, so they are moving only due to the hydrodynamic distortions. And you can see that due to the hydrodynamic fluctuations that the active carpet is exerting into the fluid, these sedimenting particles start to move and to fluctuate. And when they are getting closer to the active carpet, this fluctuation is larger. And then we can see, okay, it seems that this biogenic contribution is important, since particles really disperse around, okay? So this is an old work where the flow or the fluid, the fluid around the active carpet, it was homogeneous. So we consider a non-slip boundary here and a fluid here above, an infinite fluid, okay? An infinite fluid. Okay, but we know that in nature or inside our bodies, the fluids are not homogeneous. So we are in presence of heterogeneities or jumps in viscosities or jumps in densities inside our human tracts, inside our stomach, and so on. Then we know that we can consider that the fluid is not homogeneous, and then the easiest thing to do is to consider like two fluids of different either viscosities or densities in contact, okay? So we know that in nature, we can observe these oil slicks that probably when you observe the ocean, you can see these kind of spots in the ocean where it looks more oily, okay? Actually, it's more oily in that area, and it has been observed that bacteria accumulate underneath these slicks, okay? So therefore, we have a nice example when bacteria in aquatic environments tends to accumulate in these viscosity jumps. And this accumulation might be due to viscotaxis because microswimmers like algae or bacteria perform viscotaxis, so they are either attracted or repulsed by viscosity gradients. And it has been also observed that when microswimmers form thin films, they can also change their environment viscosity, okay? So they can accumulate, for instance, in a free surface, and since the density of microswimmers is too high, they start to change their environment in the sense that they increase the ambient viscosity around them, okay? And we can consider also change in viscosity or jumps in viscosity our inside bodies. For instance, this is an example of bacteria which is in yellow painted, and they observe that they tend to accumulate above a viscosity interface. Okay. Okay, so so now we know that microswimmers, either bacteria or microalgae, tend to accumulate at fluid interfaces. And these fluid interfaces, we can consider them as fluid viscosity jumps. So two fluids in contact with different viscosities. So this is what we will study, and I will present to you now, is we will consider an active carpet. So I will extend more in the next slide about what it is an active carpet. So for us, until now, an active carpet is a 2D film or biofilm of microswimmers, either bacteria or microalgae or dipole microswimmers. They are allowed to move only in 2D, so they are forming a monolayer, okay? The monolayer approximation is just an approximation, is to make easy the measurements, but we have observed that if we consider a thicker microswimmer's film, it is not important. It's not crucial. It doesn't change our results. Okay, so as a first approximation, we can consider that the microswimmers are forming a monolayer of microswimmers in a fluid viscous interface in our case. So the small h for us is just the size of the microswimmers. So microswimmers are actually swimming very close to the fluid-fluid interface. And we will consider an ambient fluid of viscosity mu1 above the active carpet and between the active carpet and a free surface. So here is an interface, air-liquid interface, and here we have a fluid-fluid interface. And the second fluid is below the active carpet, and we will consider this fluid of viscosity mu2. Okay, so for us, as you can see, we will have an important parameter now, which is the ratio between the two viscosities of the two fluids. And also another important parameter, it will be capital H, which is the height of the fluid film, or the height of the fluid mu1. Okay, so as you can see, this problem is a very confined problem. So we would have an active carpet in a very confined environment. Confined in the sense that is the active carpet is living or moving between two fluid interface, an air fluid interface here and a fluid-fluid interface here. So how strong is this confinement is given by lambda, which is the parameter or the viscosity jump between these two fluids. And also capital H is another measurement for confinement, since I can make H very small, so I will have a very confined carpet, or I can make H larger. Okay, so you will see that this matters too. So to solve this environment, what we did is to consider each microswimmer. So this is an example of one microswimmer here. So we will consider one microswimmer moving in this confinement, okay? So between these two fluids. And in order to solve the flow field that a single microswimmer is exerting into the flow, we have to consider an image system. So we will consider two images for each of our microswimmers. The two images are one above the free surface and the other one is below the interface. And we also are considering the far field approximation. Okay, so we are solving the flow field of each microswimmers far from their body, from their body. So the flow field is approximately something like this. It's a huge expression, so I'm only showing here a resume of the expression. You can see more details in an archive paper that we have submitted. If you look on my Scholar Google, you will find this paper under the same name of the presentation. And the important thing is that this flow field depends on where I'm measuring the flow field, so R, the distance between RS, the distance between the microswimmer and the point where I want to measure the flow field, depends on lambda, and depends on capital H. Okay. Okay. Yes. And we can still consider each microswimmer like either a pusher or a puller, but in this plot, we plotted the case of a puller. So you can see that the microswimmer, it's pulling flow into their body like this. Also, another important thing is that all our microswimmers are moving in 2D and they are moving parallel to the interface. Okay, so we consider them all moving parallel to the interface. Okay, so this is the setup. So we have here our active carpet made of microswimmers. They are uniformly distributed in positions and orientations. And from now and on, we will consider them as parallel pullers moving in 2D in this active carpet. So we will consider no hydrodynamic interactions between the microswimmers. And so we will measure the flow field generated by all the colony at the in different points above the active carpet. So we will start first considering a fluid parcel where we consider no external flow fields and that the fluid parcel is non-Brownian. And we will measure the total flow field on this fluid parcel. And if we measure that, so if we integrate or we measure the average flow field and we integrate over all the microswimmers in the active carpet that are uniformly distributed, we will measure an average velocity equal to 0. Okay? So this is not surprising for us. And then we say, okay, so the average flow field is 0, but what happens with the fluctuations? So you will see that from here and now on, in our work, we will only consider hydrodynamic fluctuations exerted by the active carpet on the fluid parcel at different heights zeta 0. And remember that we are measuring the flow field in the fluid viscosity of μ1. If we measure the variance, we will see that it's not equal to 0, or the variance is the hydrodynamic fluctuations exerted by all the microswimmers over a fluid parcel at height zeta 0. And we can measure this for different heights, and this is what we got. So we measure the variance of the velocity in terms of the hydrodynamic strengths of each microswimmer. And you can see that here in red and in green, the two variances that we are measuring. So the first one is the vertical variance. So is how fast are the fluctuations for this fluid parcel in the z direction, and how strong are them in the parallel direction, so in the plane, so in the XY plane. And you can see that first, that the hydrodynamic fluctuations are decaying in terms of zeta 0 with height. So they are larger when we are closer to the active carpet. And when we are getting farther from the active carpet, we can see that this is decay. The nice thing is that they are non-monotonically decaying, and there is clearly a dependence on zeta 0. Okay, so, and also they are anisotropic. So this means that the fluctuations, apparently the fluctuations in the z direction are larger than the parallel fluctuations above this point when they are crossing. So now we will understand what does it mean all what we are observing here. So let me show you why this result is very nice or is very important. So in our model, if you consider that lambda is infinity and also the height of the first fluid is also infinity, you can reconstruct an active carpet close to a non-slip boundary, okay? And in this case, if you measure also the variances or the hydrodynamic fluctuations, you will see a similar behavior in the sense that the vertical fluctuations are larger than parallel fluctuations, But you can see that they are also theta zero dependent, so they depend on the height at which you are measuring these fluctuations. They are anisotropic, but they are monotonous. Okay, so they are not crossing like in our case when we have confinement. So remember, this is the case only when we have an active carpet close to a non-slip boundary and we have an infinite fluid above. And in the other case, when we consider lambda equal to 0 and h to infinity, so we can reconstruct our active carpet above a free surface, we can also observe that there is the anisotropy between the two measurements, between the parallel and vertical fluctuations. We also observe that the vertical fluctuations are larger than the parallel ones. But again, we don't see this crossing point. Okay, so our question might be, what are the consequences of having this? What are the consequences on the environment fluid when we have this crossing point in the hydrodynamic fluctuations? Okay, so this is the case again of an active carpet in a fluid-fluid interface. We are considering H as 40. For us, 40 is 40 times the size of a microswimmer. And we are considering the viscous jump lambda of 1.5. Okay, so you see that we can measure theoretically where these crossings, where these two fluctuations are crossing. Okay, and after these crossing points, you can see that the parallel fluctuations are much larger than vertical fluctuations. You might be asking to yourselves if this crossing point depends on lambda or depends on h, or what is the dependency, or is this crossing point at the same height in all cases? So the answer is no. So we are showing that the crossing point is moving towards the active carpet. So remember that here in almost 0, we have the active carpet. And here, close to 40, we have the free surface. So we can see that this crossing point is moving towards the active carpet when lambda is increasing. Okay? And we are moving closer to the free surface when lambda is decreasing. Okay? So this means that if we're making lambda very, very close to 0, we will lose this crossing point, because this crossing point will move towards the free surface, like in this direction, and then we will lose it. And we will see exactly this solution here. So here I'm showing you the theoretical crossing points depending on h and lambda for our system. And you can see that the crossing point is closer to the active carpet when h is very small or the system is very confined and we have a very thin fluid. So yes. And if we are increasing h, you see that the crossing point is moving forward, is moving farther from the active carpet and moving closer to the free surface. Okay. And there is also a dependence on lambda, so the viscosity jump. And we see that when lambda is small, depending on h, we can have this crossing point either closer to the active carpet or far from the active carpet. But when we are increasing lambda, the crossing point is getting much more closer to the active carpet, which is similar to the result of a non-slip boundary. Okay. So now that we know that the hydrodynamic distortions or fluctuations are very dependent on the strength of the confinement, we can ask ourselves, so what happens if we allow small particles, non-Brownian particles, to move in this environmental fluid? Okay, so in this fluid mu1, closer to the active carpet, but not too close, since we are solving only the far field flow, right? So what we are doing now is that we will allow a small tracer particle to move in the environment, in the fluid environment. At some heights, we will start with the particles at some height zeta 0, and we allow them to move. So we will integrate the tracer dynamics or the particle dynamics only for the aerodynamic distortions. So the particle will move only due to the aerodynamic fluctuations. Okay. So in order to solve this, we have to measure it, we have to solve also in time our active carpet. And this is the way that we do it. So we are sampling an active carpet on each iteration time as a new random sample of microswimmers in different positions and orientations, like you are seeing here. So, and we are measuring the total flow field for each iteration time. And we are allowing the particle to move through this sampling. So this is what we got. So what we got is, it might be for you now very intuitive. Like if you look here and then we look here, this X, you will see that, okay, closer to the active carpet, vertical fluctuations are larger than parallel fluctuations. So tracer particles move more in the vertical than in the horizontal direction. So if we integrate a large number of tracer particles closer to the active carpet, we will see that in average they move, forming this vertical egg. Okay. Then if we go further, like in a different height, and we measure again the particle dispersion, we will expect that the particles are moving equally in the vertical and in the parallel orientation direction, and that is exactly what we observed. So this is the particle trajectories, and we observe that they are forming this very nice spherical egg. And if we go farther from the active carpet, so if we are in a height like here, we will expect that the horizontal or parallel fluctuations are larger than vertical ones. And as you observe here, we got also a more elongated egg in this direction. The eggs here are very increased in size in order to just as a schematic to show you what we are doing, but the actual results are here. And as we expect, fluctuations are also decreasing in height. So we have larger fluctuations closer to the active carpet, and they are diminishing while we are getting farther from them. So that's why this elongated shape egg is much smaller than the other two ones. Okay, so this is a very nice result. So now we can see that the active carpet in a very confined environment, it seems that is shaping their environment. So now we can measure how is what is the shape that they are giving to the environment. So we know that the active carpet or the biogenic fluctuations are shaping their environment for particle dispersion. And since particles are dispersing in the ambient fluid, we can now question ourselves, is there mixing there in this fluid or not? Like, because at low Reynolds number, it's really hard to get mixing. So this was one of our questions. So since the fluctuations are non-monotonic, monotonic, it might be, or probably this collection of microswimmers is generating mixing in their environment. So that's why we measure the velocity per correlations. So we have, again, the active carpet. So we are, again, considering the total flow field exerted by the active carpet. But now we will put at some height zeta 0 from the active carpet 2 tracer particles or 2 fluid particles. Okay, so 2 fluid parcels. And we will measure the total flow field above at these 2 fluid parcels, and we will put these 2 fluid parcels at different distance d0. So so d0, we will change d0, we will increase d0. So we will start with 2 fluid parcels very close to each other, and then we will move them further away. And what we want to measure is the per velocity correlation function. So how different is the velocity between one parcel and the other one at different distance d0, and also at different heights zeta 0 from the active carpet. And what we are measuring, we will measure the parallel velocity per correlation. So we will measure how much particles are moving in the horizontal direction or in the plane, and also how they are moving in the vertical direction, how different they are moving in their vertical direction. And these are our results. So I will explain you. So let's see first the figure A. So here we are showing the horizontal velocity per correlations at different age of the active carpet, so different thick of the film, and for different d0. So let's just focus, let's say, on the green squares plot markers, okay? So what we did is to put particles at, so we will first consider H as 20. So it's a very confined fluid, okay? And we are putting now particles at some fixed height. So the 2 particles will be here at fixed height zeta 0. And zeta 0 is 10 in this example. And now we are measuring the velocity per correlation at different distance d0. So the particles are starting very close to each other, and then we are getting farther and farther and measuring this quantity. And as you can see, we started with particles very moving very with strong correlation. So this means that when we are a small distance d0, particles are moving in the same way, or they are like very synchronized moving, okay, in the parallel direction. But if we get d0 higher and higher, or we increase the distance d0, you see that the particles start to decorrelate, okay, until we have a complete decorrelation at distance more or less 10. And 10 is the distance here is the parameter d0 over h. Okay, so this means for the case of 20 that if we have a complete decorrelation at distance d0 over h to 10, it means that d0 has to be very large, like 200 microns of distance, to get that the two particles or two fluid parcels are completely decorrelated. So we observe here that in the parallel direction, the system is super strong correlated when we have a very strong confinement. Okay? If we now measure the vertical velocity correlations, we observe a different behavior immediately. For also, let's see the case of H20. We observe the particles at close distance. They are, again, very correlated. But if we are increasing the distance d0 between them, we immediately see that they start to decorrelate, and they actually are crossing the correlation 0, and they are getting this fluctuation for negative values of the velocity per correlation. And then at some distance d0, they are completely decorrelating, and so the system is completely decorrelated. So please observe that the vertical pair correlation is now completely decorrelated. For instance, in the case of H20, at distance 40 microns. So the decorrelation is much more, it's happening at much more smaller distance than in the parallel direction. So this means that the vertical fluctuations or pair correlation is much more stronger than in the parallel direction. And also it gets negative correlation. So this means, what it means, all this, let me explain you what it means, what we are observing here. So what we are observing is that when we are very close, particles are very correlated. So they are moving in a synchronous way with the fluid. But if we are increasing the distance between them, we observe that at some point, they are also, they are decorrelating, but at some point they are correlate, but one particle is moving like up and the other particle is moving down. So they are moving in this way. And when they are moving in this way, when the 0 over h is mostly 1. So at distance, at parallel distance equal to the height of the fluid here, h, we are getting this anti-correlated motion. Okay? And this is very nice, and I will show you why. Because this is screaming that we are getting some kind of fluid structure. So apparently our active carpet is now shaping their environment again, but it's shaping now in the way that is generating coherent motion in the fluid. And it seems that this coherent motion, it's something is happening in the vertical direction. And this also coherent motion, it has a structure which is the same size of the confinement. Okay, so here we are observing that this structure is in the same size, is occurring at the same size age of our system. So what we did is to measure how strong is this anti-correlation depending on age and also depending on lambda. And what we observe is plotted here. So we observe that this flows structures that are created in the fluid here are stronger when the confinement is strong. So first, when H is small, you see that we get a higher decorrelation here. And it's not super clear how it depends on lambda. So apparently these fluid structures are appearing only due to this confinement, to the fluid thickness, okay? And lambda is also contributing, but it's contributing in the two extremes. So either when lambda is very small, so the velocity jump is very is small in the sense that when lambda is less than 1, or when lambda is larger than 2, let's say, okay? So why these fluid structures might be important, or what we are happy to observe this, is because of mixing. In this work, we didn't measure mixing because it's something harder to do, and we are looking forward to do it maybe in a next paper. But it's important because velocity pair correlations are related with pair diffusivity. And pair diffusivity is important to have an idea of how strong is the mixing process in your fluid environment. Okay, so what is pair diffusivity? So pair diffusivity, let's see this movie here. So if we have this is a movie of bacterial bath. So now we have bacteria moving in a very dense system of bacteria moving in 3D, and we disperse passive particles on this bacterial bath. And the passive particles in black circles start diffusing, and we can measure, for instance, the pair diffusivity between a pair of these particles. And what has been observed is that if we allow two particles to move not in a bacterial bath, but in a microalgae bath, we observe that the pair diffusivity collapse to the self-diffusivity when particle distance increase, which is the same thing that we are observing with the pair correlation. So particles disperse when they are farther from each other, while when they are closer to each other, they are moving coherently, and then they are not diffusing one to respect to the other one. Okay, and we can do the same with our active carpet. So now remember that our particle or tracer particles are not touching the microswimmers. They are only suspended above in the environmental fluid. And we observe the same behavior. So there is also a pair diffusivity. It's also observed that the pair diffusivity is very small at a small distance d0, and that the system is collapsing in this case to 2 times the self-diffusivity of the particles at this height zeta 0. Okay, so to finish, I will show you what we did measure, looking at this in order to observe these fluid structures that are created by the active carpet and under strong confinement. So what we did is to measure two different fluid parameters. The first one is the vertical velocity, and what we did is to paint in red in a red to blue gradient the velocities of the ambient flow here. So just to remember, the active carpet is more or less in zero, in zeta zero. And we have the free surface over here, like in 24, if I don't remember bad for this simulation. And now we are plotting what is happening in the flow environmental fluid, right? And you see that in the vertical direction, there is a strong drift towards the active carpet, but there is a strong drift here, and then there's a large fluid fluctuations over here and over here, and the fluid is almost going down, and then if you see it's going up again. So it seems that there are roll-like formations. So now we will see what is going on with the vorticity. So we measure the vorticity in the 3 different directions, and you observe that in remember that XY is the in plane vorticities, and WB zeta is omega zeta is the vertical vorticity. So what we should expect, we are expecting a vorticity in the Z direction, okay? So you observe that in the X direction, the vorticity is mostly homogeneous, like there are yellow and blue patches everywhere, so there is no clear structure there. We can see that in average, the vorticity here is probably 0. And if we look at the vorticity in the Y direction there, we will observe that here we can observe a more coherent structure. So at some point the vorticity is positive, and at the same time the vorticity is negative just by their side. And you can see that if the confinement H is more or less 20, you can see that this flow structure, when the vorticity is going in one direction and in the other direction, is more or less also 20. So it seems that this roll-like formation or these structures are it has the memory of the fluid confinement or of the system confinement, right? Which is something that we might expect if we look at other examples in environmental fluids, for example, like in bioconvection or convection and so on. And finally, the vorticity in the Z direction, again, it has a strong roll-like formation here, which is rolling in the positive direction, and you can see 2 blue formations by their sides at the same, like really, so it looks like there is a very nice structure here with a roll-like formation in one direction and in the other opposite direction immediately. And also again, all the fluid structure here, or what we are observing, is more or less on the same size of the system size confinement. So this is all what we measure. So in order to summarize, what we got is that it seems that an active carpet, which is driving hydrodynamic distortions into a fluid environment in a very confined system, where the confinement for us is an active carpet between 2 fluid interfaces. We observe that the active carpet is capable to shape their environment, so induce particle transport, which is not obvious. And also it seems that it's generating these flow structures which are larger than microswimmer size. So it's very nice. So it means that when we have a colony of microswimmers, they can generate flow structures that are much more larger than their size, okay? And I want to thank my collaborators, so my two students, Gabriela Aguayo and Felipe Barros. So Felipe Barros has performed all this investigation that I just presented to you. Is his master thesis. He's from the Universidad de Concepción. He's about to finish. Also, Gabriela Aguayo helped us to understand some stuff that appear in our in this research. He also is studying active carpets, but in presence of only a free surface. And here are my other three collaborations. Colleagues Arnold Matthijssen, Hugo Ulloa, and Rodrigo Soto, who also help us to understand what is going on in this program. So thank you very much to everyone, and good night.
Thank you, Francisca. Can you hear me clearly?
Yes.
Okay, because I just felt but for some minutes. Thank you very much for your clear presentation. We have some questions from the YouTube.
Okay.
The first one is there interest in using optical tweezers or a similar technique to study the mechanical response of this active carpet interface? Are there any groups doing that?
So there is the group of Maria Luisa Cordero in Santiago. She, with some students, are performing experiments with bacterial baths, and they are recently they are reproducing an active carpet. So in the sense that they are generating a monolayer of bacteria close to a non-slip surface, and they might be able to measure things in the fluid above this active carpet. But they don't have the technique for the optical tweezers, but we are expecting to implement it. So yeah, it's something that we are looking forward to do in order to measure like the pair correlation between two particles and so on throughout the forces in the system.
Okay, there is a comment here, Alejandro Zamorano. Thanks for your presentation. The topic is very interesting.
Thank you.
I have some questions. I was very impressed for the rows you showed at the end, because I not expect that at such sizes you could find anything similar to convection.
Yeah.
And so, but this all was on simulations, or is there any experiment in that?
Yes, it's only in simulations. Nevertheless, Maria Luisa, student who is performing an experiment with bacterial baths, they can measure vorticity in their system. So they show me that they can observe like vorticity structures also when they are confining their bacterial bath in a very confined layer. So, but the system is more or less different. So in the sense that they have a droplet of bacteria filled with bacteria, and they have inside another droplet of oil, which they can make very big in order that they have just a very small surface of bacteria covering bacteria, okay, of bacteria covering this oil droplet. And in this very strong confinement, if they measure the vorticity, they observe structures like positive and negative vorticity around the droplet. But this is a preliminary result, but it seems that it's something there. But again, it's different because they are measuring the flow field that all the bacteria is moving and all the bacteria is there also. It's not like my system when bacteria or microswimmers are not in contact with the fluid, like in direct contact with the fluid above, right? But it seems that it might appear. So we are very excited by this result. We took a lot of time to be sure that what we were looking at, it was right. But it's still not super strong, because if you see, it's not super strong, but it's there. Like you can see that it's there.
And which size are these droplets where they see this movement?
Yeah. For them, the droplets are like 100. It's more or less, the radius is like 100 microns of diameter, probably of diameter.
Okay.
And yeah, but they still have like a. Let me see if I can do it. Like so is this? No, it's like here. So is this problem here? So they have like the droplet with all the bacteria here, but they are now doing a double emulsion that they call, so they can insert an oil droplet inside here. So what they are doing is like when they insert like a very large oil droplet and all the bacteria is very confined in the border, they can observe this vorticity pattern.
So this is an oil gutlet?
Yes.
Above a suspension of bacteria?
Yes.
Okay. That was my next question.
Okay.
So is it possible to put bacteria or anything active inside a droplet and make it move?
Yes. Well, this also my colleagues have been trying to move the droplets. So it's not super easy. So they can observe that under some conditions, the bacterial droplet can like fluctuate and diffuse over a surface, but they are not really getting a very strong, like it's very a small fluctuation. It's like thermal noise. But they are looking forward to get to control the movement of the bacterial droplet. So they are trying to put so they perform experiments also with magnetotactic bacteria, and they are putting external magnetic fields in order to see if they can get or not this movement. But it's something that they are still working on.
Okay, that's really very exciting.
Yeah.
Well, let me see if we have some. No, I have no more questions. So I'd like to thank you once more for your time. And it was a very nice seminar. So thank you.
Thank you very much, Leo, for the invitations. And yeah, good night and see you next time.
See you.
Bye.